Alright, we'll get started. This is where we left off.
Like what? Like butter bread. Oh yeah. I still like it, but I'm like, if I put in something sweet, I want to have ice cream or cookies. Seriously, boba smart. I've had boba ice cream. I feel like it's almost soft. Don't want that. You can find out some restaurant, you know, 99 Ranch Market.
Yeah.
99 Ranch
This is still flowing.
No.
So we're going to compare and contrast the three different muscle types. And we have a whole chapter on the physiology of muscles. So I'm not going to teach that. I'm just going to teach you how to categorize basically what it looks like under the microscope. And so for skeletal muscle, I'll show you a picture of a muscle in the little picture there. This is a voluntary muscle. The muscles you can control with conscious thought. Muscles. When you look at it under the scope, the cells are called fibers. And they're elongated, cylindrically shaped. Sometimes they're called myocytes. Because they're the muscle. Myo is muscle and cyte is cell. They're also multinucleated. And I would say the most important thing to look for in skeletal muscle is its striped appearance or striated appearance. Striated means striped. Yeah, let's look at these features. So you see how the orientation of the fibers runs horizontally. The striations on this picture run up and down. It's very nice. Also look at the multinucleated all these nuclei. Okay, and that's it. Let's move on. This is a picture I took. Our picture is I could see the same orientation of fibers running horizontal, but then the striations go up and down in many nuclei. And yeah, that's kind of a picture I took last year. Notice cardiac, moving on to cardiac muscle, it looks very similar. One thing about the muscle fibers is they're more branched, whereas they're more parallel in skeletal muscle. And cardiac muscle, the fibers have a more branched appearance. And the branching is so that the nerve impulse can spread throughout the heart easily during a heartbeat. But cardiac muscle is not voluntary, it's involuntary. Your heart beats by itself without conscious thought. So it's involuntary muscle. I said the fibers are branched. Fibers are highly branched. To allow, I'll say, ease of signal to spread. It's also striated. And one structure you see only in the cardiac muscle, nowhere else, are those intercalated disks. Let me write it down here. Intercalated disks. These little dark bands. And what those are, they connect cells. They have desmosomes and gap junctions. Let me write that down. Desmosomes, gap junctions. Also rotation. The desmosomes, they're like a glue. So within these intercalated disks, they have a little zigzag design. You can't see it this magnification. But within there are the desmosomes and gap junctions. Any little dark band is intercalated disks. Because let's remember, the heart is a muscle. Your other skeletal muscles, that when they contract, they move joints, okay? They move your body around. But the heart's job is to pump. And so when you pump, you squeeze to generate pressure, okay? So the squeezing, when that pressure builds, you don't want the tissue to break apart, you know. And so the glue holds the tissue together. But also that spread of signal can occur through the gap junctions, okay? And because the fibers are branched, the signal will spread easily. Like for example, if you have one cell here, but it's connected to two other cells, and those two cells are connected to two other cells, and this one's connected to two more cells. If the signal starts in this cell here, and it has to spread throughout this branch system, it can do so easily because of the gap junctions. It'll spread to here, 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 and they're all branched together, so the signal will spread from one place to the other. And you'll learn about that in 431. But that's important to know for the gap junctions and the desmosomes, so it doesn't like split apart when it generates pressure. Okay. The last kind of muscle is smooth muscle. Yeah, that's a good way to think of it. Cell-to-cell communication. Oh, I'm sorry. This is a picture of cardiac muscle, although I like theirs a little bit better because you can see the intercalated discs a little more. So how are you going to tell skeletal from cardiac? The only thing is if you can tell if it's branched a little more, but that's kind of hard to tell. I always look for the intercalated discs. They're all over the place. Do you see them? I'll point to one right there. I mean, they're all over the place. You don't see those in other places, okay? All right, here's smooth muscle. So if straight, it means striped. Smooth is, well, there are no stripes. It's smooth in appearance. There's no striations. It's smooth looking, no striations. So this is muscle just like the other muscle types. The other muscle types have a striped appearance because of the overlapping filaments that make, we call them the contractile proteins. But smooth muscle has those same contractile proteins, they just don't appear striped under the microscope. And so while the other two muscles have kind of this fiber, these long, elongated fibers, smooth muscle, the cell is more spindle-shaped with a solitary nuke in the middle. So they call that a spindle-shaped cell. So when it contracts, it just kind of like shortens that way. Now if you look at the picture, smooth muscle, when the tissue grows, they stack on top of one another. So there are many layers of smooth muscle cells that we typically see in different structures. Skeletal muscle are all the muscles of your body that we'll learn about. Cardiac muscle is in one place, your heart, so that's not hard to remember. The smooth muscle, it tends to, where we see it most, it lines the tubes of your GI tract. You see smooth muscle in your blood vessels, vascular smooth muscle. You see it in other places like reproductive tract, like oviduct vas deferens. So in these structures, this isn't voluntary muscle. This is also involuntary muscle. I mean, these locations are controlled by autonomics. Let's see, all right, so involuntary. I think that's a good picture from your book. On this picture, it's the small intestine. Look for a tissue that's smooth and pink, 2 thick layers of it. Look towards the left. Do you think you see it? Smooth and pink, very dense, smooth and pink. Sometimes students confuse it for dense regular connective tissue. It is a dense tissue, but this is smooth muscle over here. There's these layers there. There is a bit of smooth muscle in other locations on this slide, but if I'm using it, I like it right here. There's actually there's a layer there. I don't know if your eye can see it. There's a layer there, and there's another layer of smooth muscle there in the intestinal wall. Throughout your digestive tract, there are 2 layers of smooth muscle. Just identify it as smooth muscle. Here's a picture of a blood vessel. It's a muscular artery. Again, I'll give you a chance to see it for yourself, and I'll point to it. Could you identify the smooth muscle? So if I say identify tissue, the pink stuff, that's smooth muscle. Do you see all these dark squiggly lines? Now that's not smooth muscle, it's elastic tissue, but I would use this primarily for the smooth pink tissue here, smooth muscle. So remember, let's go through the 4 basic tissue types. We started out with epithelial, then we did connective tissues, and today we're doing muscle. There's a 4th one that's nervous tissue, but I don't typically teach that with this chapter because again, we have a whole chapter just devoted to nervous tissue, so I'll do it later. But a common mistake students make is you forget the 4 basic tissue types. Like for example, if you were to call this cardiac tissue, I would mark it wrong. Now why is that? What's the important word? Muscle, that's the tissue type, is muscle. Cardiac describes one of those 4 basic tissue types. So cardiac tissue would be wrong. What if you just put muscle? That's wrong too. You didn't specify which of the 3 that we went over. So be careful how you identify things. Like for example, here's another picture. It's also a blood vessel. It's a nice picture. All the smooth muscle is in the vessel wall there. Okay. So if this is a blood vessel, one of those red blood cells, right? Makes sense? What they're supposed to be. You know, that concludes chapter 4. I won't cover nervous tissue. That's the end of these slides. So I'm going to go right into chapter 5 and integument. There's the classic cube O skin that we'll spend the most time talking about. You know, as a organ system, primarily the integument is skin. That's the main organ. There are other accessory structures, or we talk about with skin. For example, in this picture you can see the hair follicles, the hair root or hair follicle. In the root, there are living cells, okay, growing and dividing. Notice that the hair root, let me just finish my thought here. Okay, so you got the root, but the part we see, the hair shaft at the top, by the time hair grows out there, all the cells are dead, no living cells here. Hair shaft, dead cells. In ingrown hair is somehow this follicle gets clogged. There's a lot of other glands, for example, these sebaceous glands, which secrete an oily substance. So maybe that can clog it up and cause an ingrown hair. But we should know that also we see within the hair follicle sebaceous glands. Their secretion is oily. They give your hair that nice sheen. Okay, well, it's kind of like why we use conditioners. That's kind of your biological conditioner right there. The oily secretion from sebaceous gland. And notice how I put it under hair follicle because that's what they're associated with. So they secrete that oily substance on the surface of your hair. They give it that nice sheen. Not in this picture, but you also have nails, right? That's considered part of this system. Nails. So skin, hair, nails. Other glands like sweat glands. Sweat glands have a duct that secretes onto the surface of skin. Okay. And sweating is a very useful way to control body heat. It helps cool the body off. If you have moisture on your skin and wind evaporates it, it cools the body down. So that's part of the integument system, but there are also all these other accessory organs that are associated with it, like blood vessels. The blood vessels start at this bottom layer, and they send little branches off to the top for the gas exchange. The very top, the epidermis, is your epithelial layer. And let's remember that epithelium is avascular. It's cell-packed, and blood vessels can't penetrate them. So they go up as high as they can, right underneath them. Also notice your skin is your largest sense organ. So there's a lot of nerve endings here. So think of your skin as a big sensory organ. Because all these nerve endings, for example, let me just point to the ones that they illustrate. There's a misspelling here. Free nerve endings. You know, the free nerve endings, what does it mean by that? Well, let's just say you have a nerve ending like that. It's just free out in the open. It's a little bit easier to stimulate. And when it is stimulated by something, by some physical thing or some temperature change, you feel it. The free nerve endings convey the sense of pain and temperature, hot and cold. They also refer to these laminar corpuscles, lamellar. Now the Lamella Corpuscle, its nerve ending is encapsulated in many layers. Okay, so if you try to stimulate it, it's a little bit harder because it's got all these layers around it. And notice its location, which is more deep. The laminar, or the lamellar, or the free nerve endings, which is more deep? Just looking at the picture. The lamellar are. The free nerve endings go all the way up to here. So not only is there more layers around it, it's deeper, so it's harder to stimulate. You have to apply a deep pressure, like when you're getting a massage or you're using those handheld massagers. So when these are stimulated, they convey the sensation of deep pressure vibrations. But notice your hair follicles have nervous innervation. Hair follicle receptor. I guess it's culturally acceptable. Women shave their legs. You're shaving off your sensation, basically. That's okay, it looks good. But for example, if you're a man, you're used to having hairy legs, but you cycle, and cyclists tend to shave their legs because if they fall and get road rash, it's just easier to heal. But it's like you feel much less without hair, okay? If I blow across my hairs, I mean, I can kind of feel a little something. So they're part of the sensory function for skin, okay? Let me see if there's anything else. I'm gonna leave it out here. basic stuff.
If you were to like pull out a hair, like either like tweeze
Yeah.
a hair or whatever, are you feeling it because of that
Yeah,
ending?
that's why you feel it. Exactly.
Well, like, why aren't you feeling it by the hair follicle like receptor?
What's that?
Why aren't you feeling it by like the receptor, like hair follicle receptor? So on the photo, it looks the same as the free nerve ending, but you know, it's attached to the actual follicle.
So you're saying when you pull out your hair, this is what's picking up you pulling it out. That's why it hurts. But what were you saying?
I'm just saying, like, why isn't like that, like the one that hurts? Like, why isn't the free nerve ending?
Yeah, so these are like free nerve endings.
Okay,
I don't
just
see,
like-
yeah, there's no encapsulation around those. So they're
Just
like
a
free,
different word,
yeah.
just like attached to the thing.
Yeah, it's a free nerve ending that's
Gotcha.
associated with the hair follicle. But they just call it that since it's associated with the hair follicle. They don't call it the same thing.
Gotcha.
Yeah.
But it's the same kind of purpose.
Yeah,
Okay, just
exactly.
making sure.
Yeah, exactly. All right. So let's go through the layers. The top layer is your epidermis. The epidermis is considered a dry cutaneous membrane. With many different layers, with many different strata. That's the word they use for the different cell type layers that you see within that top layer. A dry cutaneous membrane with many different are called layers or strata. I'll go through the layers, okay? In chapter 4, we referred to it as a keratinizing stratified squamous ET. A keratinizing stratified squamous. And yeah, okay, that's what we said in chapter 4. The layer D to it is the dermal layer. So it goes to like around just to choose a boundary. Say dermis goes from here to here. The dermal layer, look at the picture. It's got all the stuff, basically. And the hair follicles there, there's the hair root. It's got the nerve endings. The blood vessels are there. In the dermal layer. Now, I forgot glands, the sweat glands. Let's put glands. Sweating is a form of excretion. Okay, so, sweating, a lot of people like to go to saunas and just sweat it out. You're trying to get rid of some of the toxins. The only other excretion function is by the kidney. Okay, when you filter your blood and you filter out the toxins. But this glandular secretion by the skin helps to sweat it out. Okay. Well, in chapter 4, we said that it's mostly a dense, irregular connective tissue. So all the collagen bundles, which are usually reddish in color, they kind of like just all go every which way down there. I'm just going to just draw it like ground beef. When you get to the top of the dermis, it has that nipply appearance. When you get to the top, it changes from a dense to more of a loose CT. So where I drew it red, that's the dense irregular. But let me change colors. How about I just switch to blue? It's more of a loose CT up here, closer to the top. That's kind of the tissue type. It's a bunch of collagen. Loose, then dense, deeper down. And that nipple-shaped appearance is important. It's how the epidermis kind of stays stuck to the dermis. Okay? They call them dermal papilla at the top. So I would note that. Dermal papilla. Papilla means nipple-shaped. Papilla. E in anatomy pluralizes it. E is plural. Okay. So the epidermis and dermis, they kind of grab onto each other because to accommodate those papilla, the epidermis has ridges to kind of fit the nipple shape. So they call them epidermal ridges, dermal papilla for a nice fit. So I add to this with epidermal ridges. So I'm going to look for a yellow pen because the hypodermis is mostly adipose. Oh yeah, yellow. So I'll just draw a bunch of like that there. I'll leave room to draw a couple of blood vessels too. Artery and vein over there. So the artery will send up a little branch, you have some gas exchange. And then the vein will take the deoxygenated blood back to the vein. This layer is the hypodermis. Hypodermis. The deepest layer. Okay, I'm going to erase some of these so I can give you some notes on hypodermis. So this is basically your fat layer. The hypodermis is, there's blood vessels. It's primarily fat with blood vessels. Okay. Primarily adipose with blood vessels. Not much else there, but you know those that fat helps keep you warm. So this kind of gets us into the functions of skin. Hypodermis, because of the fat, part of its function is thermoregulation. It can help keep you warm because of the fat. Keep warm with the adipose. But what if you want to, if the function is thermoregulation, how do you cool the body off? Sweat glands. So one, thermoregulation. Consider number one, first function of skin. Sweat glands, when they sweat, can cool down. So you got these three layers. I mean, another function, membrane barrier. Okay, part of your immune system, just a membrane, keeps stuff out of your body. Skin is a membrane barrier. And I should kind of be clear when I say skin. Skin is actually the top two layers, epidermis, dermis. Hypodermis is not truly the skin. When I skin a cadaver, I try to remove the top two layers. And in my skinning, if I see fat on what I'm removing, I'm going too deep, because that's the hypodermis. Skin is epidermis plus dermis. Hypodermis, not part of the skin. Hypodermis is fat. Also, it's interesting, when I skin, I look underneath skin. And because of the association there, skin has like a dimpled appearance. It looks all poked in there because epidermis and dermis, those dermal papilla. Okay, well, additionally, skin, I already mentioned, because of the nerve endings, is basically a big sense organ. Technically, skin is your largest organ, period. It covers your whole body. So by weight, it's the biggest organ. It's the largest sense organ as well. In terms of, I'll give you one more function here. Let me have some space here. Number 4, nutrition. Exposure to UV light helps synthesize vitamin D. Exposure to sunlight helps make vitamin D. I've spent a lot of time talking about the cube of skin. And let's look at some other pictures, some details I haven't mentioned yet. Those different strata of the epidermis, we'll overview here. That's a memory jogger. Californians like going to Santa Barbara. Let's see here. So I guess you just can't use that if you're actually in Santa Barbara, but we can say that. So CLGSB for stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, and stratum germinativum, but you could call it basali to fit the acronym. It's the bottom row. Basali means base, the bottom. So I'll go through kind of each strata so you know what to look for. What you got here is an epithelial tissue. Well, let me see if my colleague needs something. You just went to the wrong spots here. All right. Let's go. So what I did was I took a real histology picture and I just labeled it for what I saw. Okay, as you can see there. So this is our keratinizing stratified squamous epithelial tissue. Well, we're just going to define some sublayers here in chapter 5. Stratum corneum, stratum luceum, stratum granulosum, spinosum, bottom layer, stratum basale. So I did a closer up picture on this layer here so you can kind of see some of the details. To be lucid is to be clear. Don't these cells look kind of clearish to you? They're more whitish, clearish. And then granulosum, the cells appear granular. So the darker staining ones are granular. So beneath that is spinosum. The cells are supposed to have a spiky, spiny appearance. And if you look at the cytoplasm, you can see just a little bit of spikiness. Here's a picture from the book. I'll go from bottom up and describe what's in each strata. The basic idea here is you have young living cells at the bottom making keratin. By the time they get pushed to the top, they're just dead cells filled with keratin. And they're sloughed off on the top, continually replaced at the bottom. That's what's going on here. Okay. Organization of epidermis, going from the bottom to the top. So they call these epithelial cells keratinocytes because they just make keratin. Cells called keratinocytes. So young living cells at the bottom, dead by the time you get to the top. Bottom layer, stratum basale. So stratum is singular, strata plural. If you're like referring to more than one of them. So the stratum basale, these are the young cells undergoing mitosis. Young cells undergoing mitosis. These young cells, they're going to be pushed to the top. They have to replace the cells being shed at the top. There are other cells you find in this very bottom row. I tried to put a little bracket there, these bottom cells there. Melanocytes. They protect the apical surface of keratinocyte nucleus from UV damage. So you see melanocytes. Their secretion, the melanin, it's protective. Okay, it's like our sunscreen. The protective secretion. Melanin protects against UV. Ultraviolet light. That's why you get a suntan. Okay. This is basically the pigmentation of our skin. This cell type. You also have on this bottom layer, since it is the bottom layer, it can communicate with the nerve ending. You have some cells that have a tactile touch function. These tactile, they're called Merkel cells. I'll go with tactile cells. A little blue cell there is an illustration of one. Tactile cells. They communicate with a nerve ending. Communicate. With nerve ending. So this is like the sensation of fine touch. I'm blindfolded. Am I touching silk? Am I touching sandpaper? I can tell the difference. That's basically this sensation. All right, so that's the bottom layer. Some other pictures here. The illustration on the left, I'm sorry, wrong slide. This slide, yeah. The illustration on the left, you can see the extensions from the melanocyte. They're on the bottom layer, but they extend into the upper layers. And the melanin pigment gives your skin lighter or darker appearance. You usually identify them because the cell has this extensive cytoplasm around it. So here's one here, M, melanocyte. Okay. If you have darker pigmentation, it just means that these little pseudopods from the melanocyte, they're extending further up. Here's a picture from our collection, and I was just looking for it, and I was like, oh, okay. Oh, well, that cell kind of looks like it. Melanocyte right there. All right, the next row, Stratum Spinosum. Stratum physalis is basically the bottom layer, but there are 8 to 10 cell layers within Stratum Spinosum, the layer above it. For Stratum Spinosum, they called it that because the cells have this kind of spiny appearance. Spiny, I've heard described as a pincushion type of appearance. Stuff sticking out of it. They're not quite squamous with polyhedral shape. They're producing this pre-keratin protein called tonal fibrils, and that's kind of what's giving its spiky appearance. They attach to desmosomes, so they have this pincushion appearance. So cells are producing pre-keratin called tonofibrils. The layer above that, the cells start to darken a little bit. Well, anyways, there's spinosum in the picture. I just zoomed in a lot. I really wanted to give a close look at these spiny looking cells. The layer above that is a stratum granulosum. In granulosum, they have the darkest staining granules. So it's the darkest layer you see when you look at a picture. It's not as thick as spinosum. It's just 3 to 5 cell layers thick. It's the granular layer, the darkest staining layer. Cells have the darkest staining granules. So they're producing a keratin that causes this cross-linking in the cells. Whenever cells start to cross-link their proteins, it usually leads to cell death, which is what's going to happen here. So I'm writing cells produce a keratin. that causes this protein cross-linking within the cells. It will lead to cell death. Produce a keratin that causes protein cross-linking. That leads to cell death. And you want the cells on top to be dead. They're just going to be rubbed off to protect against abrasion anyway. So that's why I say in the slide, well, the cells above this layer are going to die. And also, they're too far away from the dermal capillaries. Nourishment is by diffusion. So the further up you go, the diffusion isn't going to happen as well, and cells will die. And this is kind of at the precipice of cells dying. So the layer above it is the lucidum. So there's spinosum granulosum, the darkest layer, and the lucidum right above it are just a clear rows of dead cells. So stratum lucidum, on our picture, they don't really show a clear layer of cells, so I just point to where it would be. Basali, spinosum, granulosum, lucidum would be right in there. Stratum lucidum. Clear layers of dead cells. So think of it as a transition layer between stratum, granulosum, and corneum. So there's stratum lucidum there, stratum corneum on top of it. So on this one, there's my lucidum and corneum right on top of that. Many layers of 20 to 30 layers thick. They call it a horny layer because it's not flat, right? It has all these hills on it. Stratum corneum. In anatomy, that word that means cornified, that corneum, cornified, means horned appearance. So this is the thickest layer here, 20 to 30 cells thick. The keratin cells, they're devoid of any organelles. They're just dead bags of keratin. There's glycolipids between cells, so this makes this layer very water resistant. And because it's so thick, it protects against abrasion. But it's shed continuously, but remember, it's always continuously replaced. All right, more dead cells linked together by glycolipids. For waterproofing. When you get your skin wet, it's easy to dry off. Just towel it off real quick. Also, like I said, 20 to 30 layers. That's good. You got a lot of protective layers. So the many layers protects against abrasion. Shed continuously. The fancy word for shed continuously is desquamation. Shed continuously. But the rate of desquamation will match the rate of how they're replaced. Rate of desquamation is matched by the rate of mitosis in the basal layer, so that they'll be adequately replaced. Rate of desquamation is matched by rate of mitosis in the basal layer. Stratum basale. So cells change from stratum basale to stratum corneum. It's accomplished by the specialized form of apoptosis, which is a controlled cellular suicide, where the nucleus organs break down, plasma membrane thickens, and allows cells to slough off as a kind of dandruff and dander. You shed approximately 50,000 cells every minute. So it's like I'm shedding cells, right? I mean, you can't see them, but they're falling off. Now, there is a difference between thick and thin skin that I wanted to show you with this figure. I also wanted to remind you of the dermal papilla and epidermal ridge, but I think I already mentioned that prior to this. That's how they're stapled together. There's thin skin. Thick skin, think of sole of the foot, palm. Thin skin, other places like the skin of the leg is pretty thin, but gluteal region, like very thick. Okay. Well, anyways, from here, thin skin, what we see is basically no stratum lucidum, and stratum corneum is much thinner in thin skin. Let me note that for you. Thin skin. No stratum lucidum. Thinner stratum corneum. So the organization of the dermis is arranged into loose and dense connective tissue, but they're called different things. The loose connective tissue is referred to as the dermal papilla. I think I did mention that. But where it's a dense connective tissue, a dense irregular connective tissue, I should say, it's they call that the reticular dermis. So let me note that for you. So for the epidermis, you had all those stratum to sort out. For the dermal organization, just papillary reticular. What I had said earlier, the papillary part of the dermis, loose CT, or areolar CT, which is also loose CT. Different way of saying the same idea. The reticular, your good old dense irregular CT. That's all I want you to know about the dermal organization. But because of the ridges and papilla, it does help create fingerprints Those epidermal ridges are actually create your fingerprints. So here's the papillary reticular layers. I tried to eyeball it. The tissue does look much less dense in those little pieces of dermis that stick up. And you can see a change in the density as you go from superficial to deep. Other topics that are typically taught on, I'm not going to cover all of them. I will cover burns and cleavage lines. Burns, in terms of severity, are categorized based upon how deep the burn damage goes. Whether it's fire, hot liquid, electrocution. Severity is categorized by the depth of damage. Severity of burns. So the first degree, you damage the epidermis, goes that deep. Second degree, damage to epidermis plus the dermis. That's what they're illustrating to you there. The third degree is all the way down to the hypodermis. Epidermis plus dermis plus hypodermis. If you ever see it on an exam, I also refer to the hypodermis as the superficial fascia, that fat layer. You can refer to it as either, one and the same. It's called hypodermis because that means it's deep to the dermis, but it's called superficial fascia. There's a deep fascia. The deep fascia is the connective tissue on your muscles, which is deeper. All right, so that's a term I just wanted to slide in there because it may come up again. And so the classification here, first degree, epidermal damage only, some redness, maybe some swelling, and of course pain. Lidocaine is really helpful to ease the pain of burns. Second degree, epidermal and upper dermal damage. You may have some blistering. Let's remember that there's a lot of fluid in that layer because of the blood vessels in the dermal layer. The third degree, the entire thickness of skin's involved. Skin may be gray and white, it's getting charred off. You start to lose the sense of pain because the nerve endings have been destroyed, and skin grafting is usually necessary. And burns could be caused by anything: heat, electricity, radiation, chemical burns, anything that kills the cells. Now the immediate threat, think about this, is dehydration, okay? Because the skin is waterproof, but if you take the waterproof layer away, all the blood vessels that supply blood to the dermal layers and to the epidermis, you're just oozing fluids. So the main concern in severe burn victims is fluid loss, drop in blood pressure. Put that down. Concern. Fluid loss. There's nothing keeping those fluids in, just oozing out. Make sure the blood pressure stays elevated. And so to evaluate burns, I'm not going to test you on this, I just want to mention rules of 9. We're considering the entire surface area of the body. So if you want to approximate, you kind of look and see, get a quick once over of the patient. The extent of the burns. If the entire skin surface area is 100%, we got rules of 9, so 9 for each lower extremity, 9 for both upper extremities, and the anterior trunk about 18. So you can kind of see how it breaks down. It just kind of gives clinicians, first responders, a good idea of the severity of the burn and that they should be concerned about other things like fluid loss and blood pressure. And in children, you know, those instant cup of noodles are one of the biggest causes of burns in children. And it's not so much the hot broth as it is the noodles. And also they make them in a way where it's like that. They make it so you want to spill it, right? They should make it like this, like your Yoplait yogurt. But that's not going to burn you, but that will. And so the fluid will just kind of bounce off the skin, but the noodles will stick to you. And in children, their skin is much more thin and tender, and so they burn a lot easier. So something to think about. I always serve my kids their noodles on a flat plate, just to eliminate any spill damage. So I have some stats here. It takes 3 minutes to cook ramen noodles. It only takes 3 seconds to burn them. Okay, so I have some notes there you can read. It came from the National Health and Statistics site. Another thing I want to talk about for surgeons is the idea of cleavage lines, which are kind of invisible, but be aware they're there. So if you cut along cleavage lines, you're following the flow of the orientation of the dermal collagen fibers, and it reduces scarring during healing. So it's something to consider for surgeons. Let's see here. Okay, I wanted to be sure that I covered the different kinds of sweat glands and the sebaceous glands. So these are the 3 types of glands you should know for the exam. Sweat versus sebaceous. For sebaceous glands, I already said their secretion is oily. It's a sebum. Oily sebum. It's associated with the hair follicle. That's what the picture on the right shows you. I think that's all I'll say. We already talked about the hair follicle receptors, but I just want you to know the gland. These 2 sweat glands, that one and this one, this is the Eccrine sweat gland. This is your predominant sweat gland that you see most everywhere. Sweat glands. I'll just say they're all over your skin, everywhere. And notice they're secreting sweat right onto the surface of skin. Sweat, duct. Leads to surface is what I'm writing here. You can see that from the picture too. Now the apicrine sweat gland, they're in the anal axillary regions, like your armpits, your groin region. Notice how they secrete using the hair follicle. The duct doesn't go to the surface, but it gets the sweat onto the hair follicle there. Located in axillary regions. So just think where you have your pubic hair. That's where these sweat glands are. Sweat onto hair follicle. The sweat also contains a little bit different makeup. Sweat is mostly just fluid with a little bit of salt in there. Let's see here. So the sweat in these regions, because there's a lot of hair there, the hair provides more surface area for bacteria to grow in what we commonly know that as body odor. Right? So when you put the protective, your deodorant on, usually it has something to inhibit the sweat gland secretion so you don't get body odor, or if you're sweating on a hot day. Okay. Now I'm looking at the time there, and I've decided that I'm not going to have these slides on your test, hair and nails. Honestly, I just get tired of teaching this. They're okay, hair and nails. You can look at them if you want, but I'll just say for these slides that I'm not getting to, it's not going to be on any of your exams, lab practical or lecture exam too. So I'm going to note that on the board, so you can still study them if you want. I think you might expect them on the homework at least, so you get some exposure to it, but not on your test. Not respond goal for hair nail slides. You are responsible for what I've already mentioned, so just look in your notes, but it wasn't much. These slides I'm not going to teach you. Since Wednesday's our first lab practical, I wanted to be sure that you guys kind of get the logistics of what's going to happen. So, but I have to get something for you to do. So stay put. I'm just going to run to the office and get some flashcards. I'll be back. I want to talk about the lab exam before we break.
Yeah, boba ice cream. It's a brown sugar, but it's only limited to one per customer since they sell it. Because that popular. Yeah, yeah, it's so good. Only one per customer. You know where Lauren is? It's one of these. They sell it fast because you know it's from overseas.
Серый тоже так делает, они ставят вокруг целый класс, и ты просто ходишь, и они дают.
Yeah, because they're really not popular. Not they look like this. It's so good. Yes. Sorry.
Ти будеш від першого до цієї лекції?
It's a brown sugar. Like syrup. There's no way inside. You have got to try this. I'm not kidding. It's so my sister and I love it. I'm gonna suggest it. It's so good. So good. EMT culture? It's like how mine goes. Seriously. They send their chat up, you know? $12 a box. This one we got was the best $7. Huh? But I guess it's popular. It's really popular. Thank you. Here. This is for the connective tissues.
What I need everyone to do is get a flashcard and use a big Sharpie pen. Just write your name on it. Write your name on the flashcard. I need it. So, let me just pass it. I'll just pass it back. So if you have a big Sharpie pen, fine. If not, come grab one. I want it in big black ink.